So like I said, water moves down its concentration gradient from an area of high water concentration to an area of low water concentration. So when cells are in something called as an isotonic solution, it means that the concentration of solutes is the same inside and outside the cell. Okay, and when the cell is said to be in a hypertonic solution, keep in mind hyper means higher and hypo means low. So when a cell is put in a hypertonic solution, it means that the concentration of solutes is higher outside the cell than inside the cell. When a cell is said to be in a hypotonic solution, it means that the cells have a higher that the solution has a lower concentration of solutes than the cell. Okay, now once we look at what happens to a red blood cell in these three solutions, you'll understand the concept better. Okay, so again I'm gonna repeat, water moves from an area of high water concentration to an area of low water concentration. So in other words, water moves from an area of low solute concentration to an area of higher solute concentration. So water moves from a lesser concentrated solution to a higher concentration region, so when you're talking in terms of solutes. So the first solution over here is isotonic solution. Iso means same. So the concentration of solutes over here and over here is the same. And the next picture, let me just say that this is equal concentration. The next solution is a picture of a red blood cell in a hypertonic solution. Hypertonic solution has a higher concentration of solutes than inside the cell. So initially the concentration of solutes is high over here, and low over here. Low solute to high solute. Okay, so when the concentration of solute is higher outside than inside the cell, water wants to move out of the cell. So this leaves the cell deprived of water and cell shrinks in a hypertonic solution. Now in a hypotonic solution, the concentration of solute is low outside and high concentration of solute on the inside of the cell. So water moves into the cell and the cells blow up and burst in a hypotonic solution. Now the easy way to remember this is hypo makes a hippo. So hypotonic solution leads to movement of water into the cell and the cell blows up. The cells burst. And this is the reason you always need 0.9% saline, not higher or not lower, because the concentration of solutes inside the cell is approximately 0.9, equivalent to 0.9% saline. So there's not much net movement of water. So this prevents the cells from being affected in the presence of a solution. So the other type of diffusion is known as facilitated diffusion. Facilitated diffusion, as the name suggests, requires a facilitator. This is also a process that does not require energy, but facilitated diffusion specifically transport lipid soluble molecules and larger substances via a carrier, for example glucose. Now glucose is a large molecule and it requires a carrier. It cannot just pass through the plasma membrane. So glucose is transported via facilitated diffusion using a glucose transporter. So glucose moves via a protein carrier specific for glucose. Okay, facilitated diffusion is just a process wherein the diffusion is done in the presence of a carrier because the molecules are either too big to move through or just cannot pass through the plasma membrane because of the polarity. Now, the next type of diffusion is active transport process. In an active transport process, ATP is used to move substances across a plasma membrane, and substances that are too large cannot just pass through. So sometimes they use something called as solute pumps to be pumped across the plasma membrane. Now if you remember, ATP or active transport process requires ATP, so it's like moving this piece of rock up the hill. So transport occurs against the concentration gradient, meaning from an area of low solute concentration to an area of high solute concentration. So this requires a lot of energy. So this is an energy consuming process and requires a lot of ATP. So in most cases, substances are moved against their concentration gradient. It's like packing an already stuffed suitcase and then you have to sit on the suitcase to close the suitcase. Again, an energy consuming process. So a very common example of active transport process is sodium-potassium pump. The sodium-potassium pump is necessary in all nerve impulses. So all the nerves in your body use a sodium-potassium pump to generate an electrical gradient in order to make the nerve impulse happen. Sodium is transported out of the cell in the process, and potassium is transported into the cell. Again, this is an ATP consuming process, and molecules are moved against a gradient. So this is not an easy process. So in the process of ATP transport inside and outside the cell, for every time 3 sodium ions are pumped out of the cell, 2 potassium ions are pumped into the cell. So this is a cytoplasm. So this is the inside of the cell. Extracellular fluid by default means we're talking about the fluids out of the cell. So it happens by binding of cytoplasmic sodium to the pump protein. So this is a sodium-potassium pump, which is just a giant protein molecule which makes the process happen. So the process starts when sodium ions bind to the sodium-potassium pump on the inside, and 3 sodium ions are pumped out of the cell. After 3 sodium ions are pumped out of the cell, 2 potassium ions attach to the sodium-potassium pump, and 2 potassium ions are pumped into the cell. So for every 3 sodium molecules pumped out of the cell, only 2 potassium molecules are pumped into the cell. This causes an electron gradient or an imbalance in the electrical charges inside and outside the cell. Another type of membrane transport process that requires energy is cell eating and cell drinking. So these are also active processes because they use ATP. In general, we can call them vesicular transport because they involve formation of a double membranous vesicle in which molecules are packed and transported or moved around the cell or towards the plasma membrane. So there are different types of vesicular transport. The first one is exocytosis. Exocytosis meaning when something exits the cell, and endocytosis meaning when a molecule enters a cell with the help of a vesicle. So exocytosis means something exiting the cell, endocytosis meaning something entering the cell. There are two types of endocytosis processes. First one is phagocytosis, meaning cell eating, and pinocytosis means cell drinking. So let's look at exocytosis first. Exocytosis is a mechanism when a cell uses energy to secrete hormones, mucus, and other products out of the cell. So substances are carried in a membranous sac. If you remember the earlier picture of membrane transport, there were smaller vesicles that pinch out of the Golgi apparatus and they're carried towards the plasma membrane. And these contents are emptied out of the cell. So this is a picture that shows vesicular transport. So that's the Golgi. These are the Golgi bodies that modify and package proteins into small vesicles. These vesicles are then carried towards the plasma membrane and they are spat out of the cell. So this process is known as exocytosis. So if you take a closer look at the electron micrograph of exocytosis, you can see that this is the vesicle which fuses with the plasma membrane. At the same time, it allows substances to be excreted out of the cell. You can see the things are spat out of the cell when needed. For example, antibodies, when they are synthesized, they need to be taken out of the cell. And hormones, all of these substances need to dissolve in blood to reach another part of the body. So usually they are taken by vesicles and they are transported to the plasma membrane, from where they are exocytosed out of the cell. So endocytosis, on the other hand, is something being engulfed into the cell. So endocytosis, in simple terms, you can say we have two types: cell eating and cell drinking. And this is the opposite of exocytosis. So during endocytosis, vesicles that are supposed to carry materials into the cell usually pinch off from the plasma membrane. Just say these are some chemicals that need to be carried into the cell. The plasma membrane forms enfoldings to form little vesicles, and these vesicles pinch off from the surface of the plasma membrane in the form of transport vesicles. And these transport vesicles move along the cytoskeleton of the cell and fuse into the part of the cell where these molecules need to be transported. Okay, so if they don't need to fuse with another organelle, these molecules can simply be released into the cytosol. And membrane and receptors are present on the surface of the plasma membrane. So these vesicles, when they have done their job inside the cell, can be taken back to the plasma membrane and they fuse back into the plasma membrane. So the plasma membrane does not really lose a part. It just forms vesicles that help in transport. When the vesicles have finished transporting the material, they go back to the plasma membrane and fuse with the plasma membrane. So this is a video of a bacteria being chased by a white blood cell in the body. So this is a wonderful example of endocytosis, or cell eating. So this bacteria will be chased by the white blood cell over here because this bacteria is an antigen and the cell wants to destroy it. So this white blood cell is going to chase the bacteria, eat it up, and destroy it. So let's watch the video. Okay, so that is a white blood cell chasing the bacteria. So these are spherical or cocci-shaped bacteria. Okay. The bacteria tries to surround, well, the white blood cell tries to surround the bacteria, and then we don't see the formation of vesicles, but they have been internalized by the cell, and inside the cell they will be destroyed. So with this we have finished membrane transport in the cells, and the next time or the next video or lecture we'll continue with cell division in mitosis and meiosis. So I hope you've watched all these videos before you come to class on Monday. Thank you and enjoy the long weekend.